All right, guys, we are going to start talking about Mendelian genetics. So let's just go ahead and jump straight into it. Now, has anyone ever told you that you have your mother's dimples or maybe even your father's nose? And have you ever wondered why you are a particular height, have curly hair, or maybe green eyes? All of these questions can be answered by understanding heredity, which is the passing of traits from one generation to the next. And it all started with Gregor Mendel. So, Gregor Mendel is known as the father of genetics, and he was born in 1822 in Austria. So, he became a monk and worked in the monastery gardens, and he became fascinated with pea plants that grew there. Now, Mendel's experiments with peas were able to show that genes are discrete units that keep their separate identities when passed from generation to generation. So, one of the reasons for the success of Mendel's experiments was that they were very carefully designed and controlled. So, Mendel kept detailed notes of everything that he did and what he observed. And just a sidebar, Mendel used pea plants because they were easy to grow and had many traits that were easily distinguishable, such as color, shape, height, and many more. So Mendel observed traits in his pea plants. He observed seven to be exact. Traits are distinguishing characteristics that are inherited, like eye color, leaf shape, tail length, a widow's peak, for example. Scientists knew that traits were inheritable, which means that they were passed from one generation to the next, but they didn't know how until Mendel's experiments. So Mendel observed the following traits in his pea plants. So the seven traits that he studied was flower color, seed shape, seed color, pod shape, plant height, and flower position. And he determined out of all these traits which ones were considered dominant traits and which ones were considered recessive traits. So, in genetics, we refer to the mating of two organisms as a cross. Mendel noticed that when he crossed a purebred white flowered pea plant with a purebred purple flowered pea plant, the resulting offspring looked like this. So, traits that were hidden when parental purebred flowers were crossed reappeared in the F generation. So, think about this as your grandparents' generation. This is your mom and dad, and then this is you guys. So, this is our second generation, and you can see that the white has reemerged. This is just one example of the many patterns that Mendel discovered during his experiments. Now, from these observations, Mendel drew three important conclusions, which are known as Mendel's laws: the law of segregation, the law of independent assortment, and the law of dominance. Now, organisms will inherit two copies of each gene, one from each parent. And genes are just pieces of DNA that provide instructions to make a certain protein. So, genes are what determines the traits, which are the features or characteristics that are passed on to you or inherited from your parents. Now, organisms donate only one copy of each gene in their gametes, which are their sex cells. So, here's parent 1, and here's parent 2. All right, so you can see how the cells from each parent are going to be broken down into the four cells through meiosis. But let's look at this. So, Emma's mother has a brown and red hair gene. So, she has produced two of the brown, I apologize, and two of the red. Well, over here, Emma's father only has red hair, so that's the only gene that will be found in these four cells at the end of meiosis. So keep in mind that Emma's mother and Emma's father have both passed on red hair. So Emma now has red hair because she's received that gene from both parents. This is the law of segregation. So now, before we discuss Mendel's other laws, we need to understand some important principles of heredity first. There can be different versions of genes called alleles. So you receive one from each parent, represented by letters. So, for example, you can have a capital Y for a yellow color or a lowercase y for green color in pea plants. So, this organism has a capital A and a lowercase a. Now, the alleles that an organism receives from its parents can be the same, which means that they are homozygous. But they can also give two different alleles, which are considered heterozygous. So the homozygous alleles are going to be the same. So you see, we have two capital A's here. They're going to be found on the genes. Well, for a heterozygous, the alleles are different, which is shown by the capital B and the lowercase b. Now, back to homozygous for a second. Even if it was two lowercase a's, that would still be considered homozygous because they are the same. So, a genotype refers to an organism's combination of alleles, whether it be capital B, little b, or little b. So, your genotype is what determines your phenotype. So, your phenotype is the physical characteristics or traits that an individual or an organism makes up. So, examples of a phenotype are going to be things that you can see, like if somebody has blue eyes, if the peas are smooth, if they are tall, if something has brown fur. So the genotype is the allele combinations, but the phenotype, keep in mind that pH, physical, these are things that you can see. As Mendel learned, one allele might be dominant over another. So, a dominant allele is the allele that is expressed with two different alleles or two dominant alleles are present. So, here we have a pink flamingo and we have a purple flamingo, but pink is considered dominant. So, even though there is a purple parent flamingo, because pink is dominant, our baby flamingo is going to come out pink. So the dominant alleles are represented with uppercase letters. So AA and big A, little a can have dominant alleles. Now, a recessive allele is the allele that is only expressed when two copies are present. So it's going to be little a, little a. That is the only example of something that is going to be recessive. So here we're looking at our flamingos again. So the recessive alleles are represented with the lowercase letters. So this guy over here, he's got little a, little a. And over here, this guy has either got big A, little a, or big A, big A. Which means that either genotype that this flamingo has, they have a dominant allele, which means that our baby flamingo is going to be pink because of those dominant alleles. So an organism's genotype might be homozygous dominant, which is capital T, heterozygous, which is a capital T and a lowercase t, or homozygous recessive, which is little t. And it's not just going to be T. You can do this with any letters. It is the capitalization or lack thereof that determines whether a genotype is homozygous or heterozygous. So let's talk about Punnett squares. So a Punnett square is used to show possible offspring of a genetic cross. Now, a monohybrid cross, and remember, mono, which is the beginning of this word, means one, crosses one trait between these four boxes. While a dihybrid cross, which our di means two, will cross two traits, which will give us 16 boxes. Okay, so here we have our parent alleles. We have our two eggs and our two sperms. So, what is going to be represented in the boxes are the genotypes of possible offspring. So, according to the explanation here, let's go over here to our Punnett square. So, little t, which is homozygous recessive, is coming from the mother. So, you know that whatever trait we are trying to cross, the mother is homozygous recessive, so she does not carry this. But according to Dad, he is heterozygous because he's got a big T and a little t. Now, let's look at our combinations because what you'll do is you'll bring it across and bring it down. So you'll bring the big T. So you write big T, big T. Down here, little t. And then you'll go down from the top and work your way down. So you go little t, little t, little t, little t. So looking at this, we see that we have two of the same genotypes here, which is big T, little t, which is heterozygous, just like Dad. Down here, we have two little t's, which means these are homozygous recessive, like Mom. So this child has a 50% chance of being heterozygous and a 50% chance of being homozygous recessive to this trait. Now, suppose you cross a pea plant that is homozygous dominant for purple flowers with a pea plant that is homozygous recessive for white flowers. So, what percent of the offspring will have purple flowers? What percent of the offspring will have white flowers? So, when you're trying to solve these problems, make sure that you go in and you determine what is homozygous or heterozygous and what is dominant and recessive and which phenotypes go with those genotypes. So, here we can see that homozygous dominant is a purple flower, while homozygous recessive is for white flowers. So here we have big F, which is representing our homozygous dominant purple flowers. And then we have little f, representing our homozygous recessive white flowers. So let's fill in our Punnett square. Okay, so it looks like our pea plants are going to be heterozygous. They all have a little f and a big F, which means that 100% of the offspring will have purple flowers. Because remember, the purple flowers are homozygous dominant, and all of these boxes have a big F in it, meaning that 100% will have purple flowers. So 0% are going to have white. Now let's consider a heterozygous and a heterozygous cross. So we have big F, little f, big F, little f. Here we have a different combination. We have one box that has two big F's, one box that has two little f's, and two boxes that have a big F and a little f. So here, this would be purple, 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 and white because we have two little f's here. So there is no dominant allele to turn that flower purple. So note that purple flowers are dominant to your white flower plants. Just keep that in mind. So the percentage of offspring that will have white flowers is only going to be 25% because it's only making up 25% of the box. So your phenotypic ratio is going to be 3 to 1 because you have 3 purple flowers to every white flower. And your genotype ratio is 1:2 to 1 because we have one big F, two big F, and one little f, little f. Now, how about a heterozygous and a homozygous cross? So, suppose you cross a pea plant that is heterozygous for purple flowers, which is big F, little f, with a pea plant that is homozygous recessive for white flowers. So remember what I told you. Make sure that you keep in mind that you're crossing that heterozygous purple flower with a homozygous recessive white flower. And remember that purple is dominant to white. So what percentage of the offspring will have white flowers? And what is the phenotypic ratio? All right. So the percentage of offspring that will have white flowers is 50%. Because here you filled in your two boxes that have no big F's. So you know that they are completely homozygous recessive. So your phenotypic ratio is going to be 1:1. Because it's kind of like fractions. Because we have two purple and two white. And it'd be silly to write 2:2. So we break it down to 1:1 because for every purple flower, there will be a white flower. Now hang tight with me. We're almost done, guys. We got a little bit more practice. Now describe each of the following as a genotype or a phenotype. Red hair. Red hair is a phenotype. Now big H, little h. This is a genotype. Little y, or homozygous recessive is going to be a genotype. Wrinkled peas, this is a phenotype. Freckles is also going to be a phenotype. So keep in mind, anything that is a phenotype is things that you can see. You cannot see the big H and little h in your genes, but you can see if somebody has red hair. Now describe each of the following as homozygous dominant, homozygous recessive, or heterozygous. So little y, it's going to be homozygous recessive. Big H, little h will be heterozygous. Big M is homozygous dominant. Big X, little x is heterozygous. And little g is homozygous recessive. Now, in humans, dimples are dominant to no dimples. So dimples is a capital D, while no dimples is a little d. So cross a female with no dimples with a man who is heterozygous for dimples. Tell me what is the phenotypic ratio of their offspring and what percentage of the offspring will have dimples. So we're going to put mom on top. So cross a female with no dimples. So no dimples is going to be two little d's. and a man who is heterozygous for dimples, which means they'll have a big D and a little d. Let's fill in our box here. All right. So your phenotypic ratio for their offspring is going to be 1 to 1. So for every one that can have dimples, there's the possibility of not having dimples. So 50% of the offspring can have the opportunity of having dimples because of dad's dominant allele in his heterozygous genotype. Now, in humans, freckles are dominant to no freckles. So let's cross two parents that are both heterozygous for freckles. Tell me, what is the phenotypic ratio of their offspring? What percentage of their offspring will have freckles? And solve your Punnett square. All right, so if both parents are heterozygous, they both have a big F and a little f for freckles. So let's fill in our box here. All right, so it looks like we have a dominant F in 3 of our 4 boxes. So your phenotypic ratio of the offspring is going to be 3 to 1. So for every 3 combinations that can have freckles, you'll have 1 combination without freckles, which means that 75% of the offspring will have freckles. All right, guys, that's all the practice I have for you. If you have any more questions about Mendelian genetics, make sure that you reach out, and